Okay, so our next speaker is John Murray, and he's going to be telling us about quantitative analysis of context-dependent regulation by the Wnt pathway at single-cell resolution. John?
So I want to thank the organizers for putting on this fantastic meeting and for giving me the opportunity to speak. And I also want to call out my great postdoc, Amanda Zacharias, whose work I'm going to tell you about today and wasn't able to come to the meeting. So I want to start sort of with a conundrum. We have this incredibly complicated process of development, which sort of illustrated beautifully in these movies of different model organisms, and the lower right corner there is actually a human embryo as they develop. And you have all these cell divisions, cell migrations, differentiation decisions that have to occur, across these fields of hundreds, thousands, even more cells. But this is all happening with a relatively limited, there's the human embryo hatching there, which I think is really cool. A pretty limited parts list. What I mean by that, for sure there's 1,000 or so transcription factors, of which maybe a couple hundred are used in these early patterning decisions. But the signaling pathways that are actually diversifying cell fate are much more limited, and the worm is really just sort of a couple of signaling pathways that are making most of these decisions between different cell types in early development. Right. So this is the problem of context specificity. We have these same signaling pathways that are reused for different purposes during development. So just to broaden the scope a little bit to think about other systems as well. Classically in vertebrate development, you have the Wnt signaling pathway, which is used early on in development to specify endoderm, and then later sort of both in descendants of the endoderm, as well as in other places to make other decisions, to make hindgut or to make liver, for example. So presumably, we know that this is happening largely by the Wnt pathway transcriptionally regulating different targets in these different contexts, right? And so C. elegans now is obviously a fantastic system to try and get at how this context specificity is regulated, because this Wnt signaling is being reiteratively used over and over again so often in development. Pretty much every cell division that occurs in embryonic development has differential activity of the Wnt signaling pathway as measured by the concentrations of its vector proteins in the nucleus. Right. And we have this invariant lineage, a beautiful invariant lineage that comes out at the end of that, where we can take advantage of this to map the activity of the pathway and the expression of targets across all of the cells. Right. So we, many years ago now, with Zhirong Bao and Bob Waterston, developed this approach using confocal imaging to track all of the cells of development, and then automatically, by using cell recognition, cell tracking software, reconstruct the lineage for the entire organism. And by expressing in the same embryo a second color reporter, we can use that to map gene expression. In this case, we have a red fluorescent protein that's driven by a promoter for the FA4 gene, and these embryos then go on to elongate and hatch normally. So the output of that is something like this, where we have this lineage tree now here at the bottom, with this color-coded in this funny heat map scale, which I'm going to keep using throughout the talk, where purple is no expression, and then as you get to green, blue, green, yellow, red, you get progressively higher levels of this nuclear concentration of the reporter in every cell of the embryo. And for these kind of transcriptional reporters, I think one thing that will become important later is that there's a little bit of a lag. So when you see these expression in these clonal lineages, we infer based on the time that it takes for transcription, translation, and fluorescent protein maturation that the actual initiation of transcription or expression happened, the decision to express these genes happened up here in this founder branch. All right, so brief primer here on the Wnt pathway in C. elegans. So this is happening in, again, almost every cell division. This is work from many labs that established this over the last 20 or so years. So you have a cell that's most of the cells divide along the anterior-posterior axis. You have Wnt ligand interacting with this receptor that orients that division that way. And through a series of signal transduction events, the beta-catenin protein, cis1, enters the nucleus of the posterior cell, which we'll sort of, by shorthand, call the signaled daughter even though the signaling is actually happening during mitosis here. Also, this Nemo-like kinase protein, there in yellow, enters the nucleus as well, phosphorylates the sequence-specific transcription factor, TCF, or POP1, cause it to be exported partially from the nucleus. And the end result of this is that you have this stoichiometry difference between these two proteins in the anterior and posterior sister cells, such that in the anterior cell, TCF or POP1 is bound to its targets primarily without beta-catenin, and so acts as a repressor. And in the posterior daughter, it's bound instead as a complex with the beta-catenin, which is a co-activator and drives active expression. All right, so we use that lineage approach that I just showed you 2 slides ago with the movies to ask which cells have high nuclear beta-catenin. And we were thinking this is sort of a control experiment, because this has been worked out in the early lineages pretty well, and by many labs using sort of lower throughput approaches. And so which cells have high nuclear beta-catenin? You can see in this, based on what I told you about the lineage, every cell, when you have a division, you have higher nuclear beta-catenin in the posterior daughter. And so it's not surprising you see high levels of nuclear beta-catenin. This is using a reporter that was generated by Judith Kimball's lab that rescues the null mutation in cis1, I should say. So you have high levels sort of throughout the animal. So even though the Wnt ligand, which I'm not going to talk much about Wnt ligand, comes from the posterior, somehow the signal is being relayed across the entire animal. So it's not really a strong positional bias in beta-catenin. And you see this sort of this incredibly striking pattern where in the lineage, where every time a cell divides, the posterior daughter has a high nuclear beta-catenin, like what was predicted from before. But what was interesting about this, so we have here's this lineage here is a more anteriorly located, a more posteriorly located lineage in the embryo. They have basically the same levels. If anything, there's a little bit more expression towards the anterior part or more nuclear localization in the anterior part of the animal. But there's this lineage sort of history effect, which we found to be really striking. And basically the idea here is that when you have a division, if your mother also had high nuclear beta-catenin, then the posterior daughters of those divisions have more nuclear beta-catenin than this cousin lineage, where that's the daughter of an unsignaled cell. And this is something that continues to propagate through development. So we were intrigued by this, and so it suggests there's sort of a memory of previous signaling events that's happening as these cells are dividing. You can do the same kind of analysis. Now this is again a chunk of lineage with that same beta-catenin reporter I showed you before, and this is the reporter for the TCF transcription factor, POP1. Remember, that's highest in the nucleus of the anterior and unsignaled daughter cells. And you actually see the same kind of a cousin enrichment going on with that protein as well, where the doubly unsignaled cells seem to reliably have more nuclear TCF, at least for this reporter, than their singly unsignaled cousin cells. And we actually went and validated for some lineages with a POP1 antibody to show this actually happens with the endogenous protein. All right, so if you combine this across the entire lineage, we can actually sort of break down the cells into sort of 4 categories based on whether they were signaled once, signaled twice, or unsignaled once or twice, and plot those here based on the concentration of their beta-catenin and TCF in the nucleus. And you can see there's these 4 groups depending on the history of division state. And basically, are you a single posterior, double posterior, or single or double anterior cell? They give these relatively consistent slopes that sort of suggest that these are, you can think of these as sort of like the stoichiometry between these 2 proteins. So, in addition to that, I don't want to diminish the fact that there's this large variability in the overall levels of both proteins across cells, and some of that has to do with time and development. I don't want to go into it too much, but there's other contributors besides just this lineage effect to the levels of these proteins. Okay, so we wanted to know, is this affecting gene target expression? So to get at this, we went to the EPIC database, which is a resource we generated of wild type expression patterns for different reporters using the same lineaging approach. This is an example of just four well-studied genes and showing how you can collect these lineages for different embryos and then merge them onto a reference lineage to look at co-expression. Right? And the current version of this now in the Waterston lab has over 250 transcription factors that they have these data for. So I encourage you to check this out and look for your genes of interest on there. So we mine this for looking for genes that are expressed specifically in posterior daughter-derived lineages, which might be Wnt targets. So here's some examples of those. These are all genes expressed in those posterior lineages where this is where beta-catenin is highest. And we found so it's confirming these are, we don't know if they're direct or indirect at this point, but these are targets of Wnt pathway based on the fact that if you get rid of POP1, you see changes in gene expression. So here's a classic example of this, the END1 gene, where remember the TCF is both a repressor in this lineage and an activator in this lineage. And so if you get rid of the TCF in this case, you see de-repression here and also some deactivation there. But actually, most of the other targets we found only had one of these two behaviors. So here's an example, NOB1, where if you get rid of both TCF or either TCF or beta-catenin, you lose expression in these posterior lineages. So this is a target that requires TCF for activation. Here's a different target, say 36, which seems to require TCF for repression and doesn't really mind that much if you get rid of beta-catenin. All right, so how is this working? So the model is that you have basically, the way to think about this is if you think of the regulatory elements controlling a gene, what's the activity of those without the TCF binding site would be the effect of the context factors, right? And so some genes might be perched right at the boundary there, in which case you'll see phenotypes, basically TCF is having a role both in activation and repression, but most of the genes that we identified looks like they are probably, the context sites are either not sufficient at all for expression, and so you need the activity of the TCF to turn them on, or they're sort of constitutively on in whatever lineage, and you need the activity of TCF to turn them off in the unsignaled lineage. Yeah. All right. So if this quantitative effect that I showed you is important for these genes' regulation, we have two quick predictions here. One is that the genes that are expressed in these multiply signaled lineages where the activation is highest should be more likely to fall in this class where they require TCF for the activation. The second is that changing the amount of TCF binding should change the expression in multiply signaled lineages. Okay, so this is just, I'm going to quickly go through this to show you these are both true. So this is the genes sorted by how much of their expression is in these doubly signaled cells, and then color-coded by which class they are. And so this matches our prediction. These genes that are expressed in these doubly signaled lineages are much more likely to require TCF for activation. Okay, to get at the second question, we use synthetic enhancers. And this is basically taking 7 TCF binding sites, putting them next to a minimal promoter. You can see there's expression in various lineages, some of which are singly, doubly, or triply signaled lineages. And as we remove TCF sites and go to 6 or 3 sites, you progressively restrict expression to primarily double posterior, or even in this case, triple and quadruple posterior lineages. Right, so these seem like they're consistent again with the model. These things are important. So I think sort of the final model here is that in addition to the context inputs, the classically would be determined other transcription factors to bind to these enhancers. The actual activity of TCF and beta-catenin in the cells is actually sort of tuning this range in which the signaling can boost expression above or below the threshold that's needed for expression. And I think that may be another mechanism that contributes to context. Okay, so I told you we don't know yet for any of these targets if they're direct or indirect. We do know that many of them have enhancers that seem to drive similar patterns, and they're highly enriched for TCF binding sites. So that suggests that many of them are likely to be direct. There's just one example of that where you can see the full promoter and one of these enhancer regions that we've cloned. So far, we've tested 14 enhancer candidates for these targets, and 11 of them are driving posterior lineage-specific expression, and we're continuing to do more of this. Okay, so just to summarize, we have this model that we're going to want to propose, and maybe there's some speculation that Wnt in this system is acting sort of as a transmitotic morphogen, which is a term that Joel Rothman, if he's here, introduced to us. Basically, as opposed to a classic morphogen where you have the sort of diffusion over a long space of changing activity spatially, this is now the same signal that's being applied reiteratively as the cell divides and having a different effect depending on how many times it's been applied. I didn't show you, again, more data that this requires. We have some idea about how this happens, but we still don't have a really good handle on the mechanism. Right now, all we know is it requires the Wnt receptor and proteasome. And we think this is maybe integrating with some of these other lineage-specific transcription factors to ensure correct expression. So, sorry I went over the time for questions here, but I just want to thank the people in my lab, and especially Amanda Zacharias, who did all the work that I talked to you about, with some help from Travis Walton, who's a lab alumnus now. And Amanda's on the job market this year, and so I encourage you to look at her application as it comes to you. And thank you.